Good morning, dear students. Hope you are doing good. Today, I am going to take the topic on Mycobacterium tuberculosis, especially the pathogenesis of pulmonary tuberculosis. As an introduction, this Mycobacterium tuberculosis comes under the family Mycobacteriaceae and order Actinomycetes. General properties of the bacteria include acid fastness, that is, they resist decolorization by dilute mineral acids. Acid fastness of this bacteria is due to the presence of high content of mycolic acids in cell wall, and also this acid fastness is due to the integrity of cell wall. These organisms are non-motile, non-sporing, non-capsulated, weakly gram-positive, straight or slightly curved rod-shaped bacteria. These are obligate aerobes. Some are microaerophilic, occasionally branching filamentous forms resembling fungal mycelium can be seen. So coming to the history, this leprae bacillus was discovered by Armor Hansen, hence it is called Hansen's bacilli. Robert Koch isolated the tubercle bacilli, that is Mycobacterium tuberculosis. So coming to the classification, this Mycobacterium tuberculosis comes under the classification Mycobacterium tuberculosis complex, and Mycobacterium tuberculosis causes tuberculosis in man. Mycobacterium leprae causes leprosy. Non-tuberculous mycobacteria, these cause opportunistic human infections. Then saprophytic mycobacteria can be isolated from soil, water, etc. So this Mycobacterium tuberculosis complex consists of Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium capreae, Mycobacterium microti, Mycobacterium africanum. So coming to the virulence factors, these organisms contain antigenic structures, that is cell wall antigens. So cell wall antigens that consist of cell wall layers, peptidoglycan, arabinogalactan, mycolic acid layers. Peptidoglycan layer which gives shape and rigidity of cell wall. Arabinogalactan layer helps in survival within macrophages. Mycolic acid layer confers very low permeability and responsible for acid fastness and reduces entry of antibiotics. Outermost layer of this organism consists of lipids, glycolipids and mycosites. Proteins present are transport proteins and porine proteins. Plasma membrane is present beneath the cell wall and it contains antigens like lipoarabinomannan, phosphatidylinositol monocytes and proteins. Then another class of antigens are cytoplasmic antigens that are responsible or that helps in the zero diagnosis of this disease. So this is the diagram representing the cell wall structure of Mycobacterium tuberculosis. So coming to our topic, that is pathogenesis of pulmonary tuberculosis. The source of this infection include humans, and for humans the disease condition is pulmonary tuberculosis, and bovine tuberculosis is due to the consumption of unpasteurized infected milk. Mode of transmission of this infection is by inhalation of droplet nuclei, that is while coughing, sneezing or speaking of infected patients, ingestion of contaminated milk or swallowing of sputum in case of infants, inoculation through that skin to skin contact, and it is a rare mode of transmission, that is inoculation. So we can simply call it as 3I, that is inhalation, ingestion and inoculation. So what are the risk factors that helps in the pathogenesis of this disease condition? Low cell mediated immune response in people like HIV infected people, then bacillary load, then overcrowding in poorly ventilated room, age, transplantation, trauma, surgery, drug abuse, smoking, etc. So there are certain sequence of pathogenic events that takes place during this infection, that is When we inhale the droplet nuclei containing tubercle bacilli, this tubercle bacilli from the infected patients reach the alveoli, and the next step in the sequence of pathogenic event is addition to macrophages. That is, mycobacterial surface lipoarabinomenin binds to the complement receptors and mannose receptors present on the surface of macrophages, and this leads to the internalization of bacilli. Next step is phagocytosis by the macrophage, that is enhanced by C3b complement mediated opsonization of bacilli. After the addition to macrophages, survival inside the macrophages, that is multiplication inside macrophages by resisting the phagosome-lysosome fusion by bacterial cell wall lipoarabinomenin, thereby inhibiting increase in intracellular calcium and phosphatidylinositol 3-phosphate level. So bacilli successful in arresting phagolysosome fusion, and they happily replicate inside the macrophages. So what happens? After that, this infected macrophages eventually ruptures and release its bacillary contents which infect other phagocytes, and the cycle continues. So these are the sequence of pathogenic events that take place during the pulmonary tuberculosis. So this is the figure showing the pathogenesis of Mycobacterium tuberculosis. In this step 1, these organisms get adhered to the macrophages. After that, internalization of bacilli, so multiplication of bacilli in macrophages, and this infected macrophages will rupture and release the bacilli which infect the neighboring macrophages. So what are the host immune response responsible for this kind of infection? That is, tubercle bacilli are engulfed by macrophages, but they survive and multiply in macrophages. They lyse the host cell, infect other macrophages, and sometimes disseminate to other part of lung and elsewhere in the body. Cell-mediated immunity plays a major role to interact with these macrophages, whereas humoral immunity appears to be irrelevant. CD4 plus helper T cells secrete interferon gamma, interleukin, tumor necrosis factor alpha, and others, exerting different biological effects. So it may result in protective immunity or delayed type hypersensitivity reaction. So T helper 1 dependent cytokines activate macrophages to kill intracellular mycobacteria and thus result in protective immunity. T helper cell 2 cytokines induce delayed type hypersensitivity, tissue destruction, and progressive diseases. So this is the host immune response involved in the pathogenesis of this pulmonary tuberculosis. So this Mycobacterium tuberculosis that comes under the family Mycobacteriaceae. It's an acid-fast bacilli. So due to the presence of this mycolic acid layer, they show acid fastness and also shows high integrity. So coming to the cell wall structure, cell wall antigens, there are 2 types of antigens, cell wall antigens and cytoplasmic antigens. So after that, pathogenesis, mode of transmission is by inhalation, ingestion and inoculation, and the source of infections are humans, and for humans the disease condition is pulmonary tuberculosis, and bovine tuberculosis is there, that is due to the consumption of unpasteurized milk. After that, there is a sequence of reactions during the pathogenesis of these conditions, that is adhesion to macrophages, internalization of bacilli inside the macrophages. Then these bacilli, after entering the macrophages, they resist phagosome-lysosome fusion, resist phagocytosis. This bacilli gets multiplied inside the macrophages, and after that the macrophage rupture and infect the surrounding macrophages, and they form a multinucleated giant cell called gone focus or granulomas. Okay, so this is the pathogenesis of pulmonary tuberculosis. So hope all of you understand the topic. So we will come with another topic, that is the clinical manifestation, all in the next section. Thank you.